we get experimental science. We're curious, non-judgmental.
Alrighty, y'all. Songbirder guessed it. It is in fact a cicada. There are a whole bunch of cicadas over in Florida, so I thought it'd be kind of cool to bring one back. A dried specimen that we identified. I really like the shading of this. Yeah, it looks really, really nice. This is a relative of one of those hoppers that we identified. How big is the specimen? Ubi, this is quite a small cicada. This is about 4 centimeters in length. There we go. See, each one of those circles is an eye cell. It has a neuron that runs deeper into the brain. So yes, as Miss Yvette points out, there are hairs around the eye of this animal, but I also find striking just all those little cells there. And remember, those connect deep into the brain and will be pulled together in what is called the optic lobe, the lobula, and then it makes an image together. What shape are the eye cells if we can get closer? It's not standard spikes amongst all insects, so honestly we'd probably need to have a scanning electron microscope. There's also ones where you do have a circular effect, so there's little rims that are touching, and the reason it's circular, and there's gaps in between each circle, and there's little sensory bristles that stick out of it, and we just can't get that zoom here, and some are much more hexagonal in shape. But even those aren't necessarily interlocking. There are sensory bristles. Where you'll see differences are along the rim. So along the bottom down here, you'll see differences in the cell types. Not necessarily the cell types, but the shape, just because they're terminating at this edge. But if you were just to take an image here in a scanning electron micrograph, they'd all look the same. They're pretty uniform on that front. The reason for that is from a developmental biology perspective, it's just easier to program having all the same cell type and appearance instead of having a unique and different one for each type. The sensory bristle spikes are in part, we think, for cleanliness, so that they can clean their eyes without scraping the retina. There are mutations that you can only study with a scanning electron microscope, and what ends up happening is that those hairs don't form uniformly, and then they have visual problems. What's not clear, though, is like chicken or egg question. Like if they have vision problems because they clean their eye in a weird way, or because it's something else unrelated. So you get images like this, and you can see the difference in those sensory bristle patterns. So that's wild type here, and then you have mutant ones here, and you can see now the hair bristles are pointing all over instead of uniform, and there's those cells as well. So this is much more circular for the fruit fly, and in between those little gaps you have those sensory bristles coming out. Yeah, no eyelids, no tear ducts. These are not damp. Their retinas are not wet. Totally dry, in fact. Like regular part of the exoskeleton. There's a membrane underneath here that prevents water from escaping, and so everything underneath is able to stay damp. And then here are some of the mutations that cause some of these weird effects. This one in panel D corrects a lot of it, it looks like. Did y'all know what these are? Any guesses? Warts? No, Smikes! Not quite the warts. Not antennae, Miss Yvette. Quoth the Raven is correct. These are the ocelli. These are what are termed the false eyes. They're usually on top of flying insects' heads, and they detect UV light. And so it's a directional eye, as it were. You can see there's 3 of them. There's 1, 2 right now in the field of focus, and there's another one back there. So it's those 3 that they're utilizing to basically to have a sense of direction when they're flying just during the day. So this is the thorax. See all the sensory bristles on the animal as well. None of these, I look at it and I can't tell with my naked eye that these are even there. So it's really cool to be able to see underneath the microscope. And then here are the tergites. They're quite visible in this section here. Each one of these sections, 1, 2, 3, 4, those are tergites. And those allow the abdomen to expand and contract. I'm not sure if this is a male or a female, but with females, their ovaries expand and shrink with respect to food intake. And so that also allows them to change the size of their abdomen based on that fat content size. If it was all one solid piece, which is usually what I think that these insects look like, just a solid piece of armor, then there's no expansion or contraction. There is just a single type of body plan. Trying to identify where the noise-making organ would be, but it's going to be right underneath, between the thorax and underneath the wing. Cliff, unfortunately, if I move this wing, it will break the sample, and I think it's a really, really pretty sample, and I don't want to crack it. She has her wings pointed down, so it's able to keep it look very nice. Cliff, that'll be right underneath, I think, where the wing is blocking off. There's a little noisemaker. right there. That's what we were looking for. We'd have to pull up the wings, but given, again, both wings are down by her side and how dried out she is, I don't think we'd be able to do it without breaking her. But yeah, so this one, right, Cliff, looks like it has those same little curly bits. Quoth, I think, my suspicion tends to go to female, but let's see if we can actually ID her. I was explaining the concept of the tergites in the context of this being a female because that's where the most important changes can occur. There can be an increase in abdominal size that connects to the ovary size. So the ovaries respond to nutrition. They don't have a set number of ovaries, eggs number. Instead, they're dependent on food intake. So if they have low amount of food, their ovaries shrink. And if they have a high amount of food, the ovaries grow. And they need to be able to expand that within the abdomen. That's why they have those tergites. The females make sounds to only the males. I think corn. I've read both. I think it's species dependent, though. I think it's species dependent, but I have seen examples where both. And actually, corn, that's a unique and cool discovery because what was identified was we thought that a lot of insects, it's only one directional, where it's only the males do the singing. I don't want to make a general comment, Spikes, because it's probably going to be there's an exception to the rule, but I have a suspicion that that's a pretty good rule of thumb. I don't know if there's violators to that rule. I imagine there are, just because there always is. But as far as I know, that is in fact the case. That there is that subcutaneous layer, especially due to the reproductive organs. Quoth wanted to analyze if this is female. This is female. That is an ovipositor right there, Quoth the Raven. So we do in fact have a female here. So fallopian tube, Smikes, is the tube where the embryos develop. However, in insects, there is a ovarian duct. Above it is where the eggs develop, and then the duct is where they're laid. It looks structurally like the fallopian tube if we were to draw it out, but the embryos don't develop in the oviduct. They're just fully developed at that point, and then the eggs are deposited and laid. Apparently, the ovipositor of that species has like a tungsten and other metallic elements on it so it can penetrate trees. Yeah, Neuro, what's wild to me too is that there's some that have like their proboscis or their mouth part, and that's where they can penetrate trees and drink out the xylem and the phloem. Thought embryos developed in the uterus. That's true, Smikes. There is some development that takes place in the fallopian tubes because that's where fertilization takes place. So you have some of the initial developmental stages happening there, and then it goes to the uterus to implant.